DNA replication is pretty efficient, and out of 3 billion base pairs, only a few mistakes are made each time it divides. But DNA can also be damaged by things like ultraviolet light, X-rays, reactive oxygen species, or chemical mutagens. So how bad is a DNA mutation? Well, that depends on what type of mutation it is. I already talked about silent mutations due to the third or wobble base in a codon. Here's the same example as before, where you can see that even if you change one of these third bases, you still get the same amino acid. So this is the least problematic kind because it doesn't really have any effect on the protein. A missense mutation is also caused by changing one nucleotide, but in this case it will also change the amino acid that the codon codes for. In this example, we're changing the second base from CCU to CUU, which makes an amino acid change from proline to phenylalanine. This can be relatively harmless or extremely harmful, depending on a few things. First of all, if the mutation is in an important part of the protein, such as its active site, even a single amino acid change can prevent some enzymes from functioning. Second, changing from one amino acid to a similar one, such as from one medium-sized nonpolar amino acid to another medium-sized nonpolar amino acid, will usually have a smaller effect than changing from a positively charged to a negatively charged amino acid, or vice versa. Lastly, the severity of the mutation depends on the importance of the protein, since some proteins have more vital functions than others. But generally speaking, missense mutations are usually not that big of a deal. Next is a nonsense mutation, which is when the nucleotide change converts a codon that codes for an amino acid to one that codes for a stop codon. If we look at our codon table, you can see that UUA usually codes for leucine. But if that middle nucleotide is mutated to become an A, then we get UAA, which is a stop codon. Nonsense mutations have a much stronger effect on protein function, since you lose everything downstream from where the mutation occurred, which often has the same effect as deleting the protein entirely. Okay, so everything I talked about so far has been changing one nucleotide base into a different one. The frameshift mutation is unique among all the ones listed here, because it's the only one in which nucleotides are added or removed, rather than just changed. Since this throws off the anomalous, non-overlapping code that I mentioned a few minutes ago, everything downstream from the mutation will code for the wrong amino acids. For example, if we have this sequence broken down into these codons by color, we have the sentence, "Can you dig her new hat?" But if we add the letter X right here, all these codons would be shifted over and will now code for something different. So unlike a missense mutation, where you have one different amino acid, now you'll have a lot of different amino acids. Frameshift mutations usually cause a similar amount of damage as nonsense mutations, but in some cases can be dramatically worse. Can you think of why? Nonsense mutations usually just result in a less effective or non-functional protein, whereas frameshift mutations result in a new amino acid sequence, which can take on a new function, and these new functions can really throw off cellular homeostasis, potentially leading to cancer or genetic diseases. And in fact, two diseases that are often caused by frameshift mutations are Tay-Sachs and familial hypercholesterolemia. The last type of mutation I want to talk about is a splice site mutation. This is exactly what it sounds like, a mutation at a splice site. We'll go through splicing in more detail later, and if you want to skip ahead and watch that video before this, you can, but I'll summarize it here. When a gene is transcribed into RNA, the RNA first includes both introns and exons. Which ones are removed? That would be the introns, which I remember by thinking that the introns stay in the nucleus, since they're removed, while everything else goes out into the cytoplasm to be translated into proteins, like this one here. But what if we have a mutation right here at this red star? The splicing machinery needs to recognize very specific sequences to know where to splice. So if one of these sequences has a mutation in it, splicing won't happen. And the whole RNA will head out into the cytoplasm together from the nucleus and be translated into a protein. But now the protein might have another section in it that it didn't have before, since the ribosome tried to read the intronic sequence as well. So how severe is this type of mutation? Well, it really depends. For the most part, this will just result in a non-functional protein, either because the extra part prevents it from functioning normally, or because some of the intronic sequence had a stop codon in it, and we end up with only part of a protein being translated. But it's also possible that we could create a new function for this protein, which could have very serious consequences, such as cancer. The lac operon is a genetic mechanism used by bacteria to decide whether they should either use glucose or lactose to make ATP. So why do you have to know about the lac operon? Well, honestly, this is something that has no clinical utility by itself, but is taught and tested because it's a classic example of gene regulation, and the same principles apply to tons of other things that are more relevant to medicine. Okay, so how does this work? Well, first, it's important to understand why the cell cares what substrate it's using in the first place, which comes down to energy efficiency. The cell would really prefer to use glucose, but it's okay to use lactose instead if no glucose is available. A key point here is that the cell needs to expend some energy to produce and maintain the enzymes and machinery required to metabolize lactose. So if glucose is available, then there's no need to synthesize these lactose metabolizing enzymes. That's where the lac operon comes in. It senses how much glucose and lactose are available and uses that information to decide whether or not to produce the lactose metabolizing enzymes. Okay, let's go through some examples. If both glucose and lactose are available, does the cell want to synthesize the enzymes that metabolize lactose? No, because remember, the cell prefers to use glucose, so if both sugars are available, it doesn't really care about the lactose and won't waste energy making the enzymes needed to metabolize it, since it's happy using glucose alone. There will be very low expression of the lac enzymes, and I'll explain why that's the case later. Let's do another one. What if there's no glucose but there is lactose? In this case, the cell wants to make these lactose metabolizing enzymes because only lactose is available, so this is what you're going to use to make ATP. Next, hopefully these are getting easier now. If you have glucose and no lactose, do you want to make the lac enzymes? No, because you have glucose and there's no lactose around to metabolize anyway. And lastly, if neither glucose nor lactose is available, what do you want to do? Well, again, there's no lactose available, so it would be a waste of resources to make the enzyme needed to metabolize it. Alright, now let's go over the molecular interactions that allow the cell to make these decisions. This gray bar is DNA, and the genes needed to metabolize lactose are these: LAC-Z, LAC-Y, and LAC-A. And upstream from these genes is where we'll regulate whether or not we want to transcribe them into mRNA and then make the proteins that correspond to these genes. An important point here is that since this is a prokaryotic system, we can make polycistronic RNA. Do you remember what that means? Polycistronic mRNA has more than one gene on it, so in one continuous round of transcription, we make an mRNA molecule that has these 3 different genes on it. This does not happen in eukaryotes like us. Now let's walk through how we get transcription. When do we want to transcribe these genes? When there's no glucose and plenty of lactose. So, when glucose is unavailable, first the enzyme adenylyl cyclase converts ATP into cyclic AMP. Cyclic AMP can then bind to an activator protein called catabolite activator protein, or CAP, causing it to bind near the promoter and help recruit RNA polymerase to transcribe the genes. But wait, we only want to transcribe these genes if we have lactose, right? So how do we make sure we don't transcribe them if there's no lactose? Well, we have this inhibitor protein, which is encoded elsewhere in the genome by the gene LAC-I, where the I stands for inhibitor. This protein binds to a region called the operator and prevents RNA polymerase from transcribing the LAC genes. However, if lactose is available, it binds to and inhibits this inhibitor, allowing transcription to proceed. So if we have lactose, we can make the proteins that correspond to these 3 genes, which are needed for lactose metabolism. Now what about glucose? Well, if glucose is present, we don't want to transcribe these genes. So the enzyme adenylyl cyclase has a binding site for glucose where glucose can shut it down, so the CAP protein will never bind to the CAP site or recruit RNA polymerase to the promoter. Now this particular mechanism isn't 100% effective. As I mentioned earlier, if you have both lactose and glucose, the LAC-I protein won't bind to the operator, and CAP won't be activated. But you will still get a very low level of transcription of these genes. On the other hand, if you don't have lactose, you get pretty much 0 transcription because the inhibitor protein is a lot more reliable. Alright, so let's say we have some of these mutations and we want to fix them. How can we do this? Well, it depends on the type of mutation. Ultraviolet light, which is the part of sunlight that gives you a sunburn, causes adjacent pyrimidine bases in DNA, mostly thymidine, to covalently attach to one another, forming a dimer. This can cause mutations in the next round of DNA replication. This is the most important kind of mutation that is resolved by our first repair mechanism, called nucleotide excision repair. Basically, the enzymes involved in this repair pathway will cut the damaged strand upstream and downstream from the site of the damage, and remove the oligonucleotide, and then DNA polymerase can come fill in the gap and ligase seals it shut, which is kind of like what we saw in the DNA replication slide. If the enzymes involved in this repair pathway are mutant or deficient, this causes a disease called xeroderma pigmentosum. This is a really serious disease, and people who have it rarely live past the age of 20. Cells in these patients can't repair pyrimidine dimers, so they're particularly sensitive to sunlight and need to avoid it completely if possible, and they usually die of melanoma or squamous cell carcinoma. Here's a picture of squamous cell carcinoma in one patient. Base excision repair is a second type of repair mechanism which fixes bases that have been covalently modified, and it starts just the way it sounds. The damaged base is excised after being recognized by glycosylases. So let's say you have a stretch of DNA like this. and maybe this cytosine gets deaminated. To repair it, the glycosylase that's specific for that base, which is cytosine in this case, will come over and remove just the base. This results in an apurinic or apyrimidinic site, or AP site, and either just that single nucleotide is removed, which means that the purine or pyrimidine base has been removed. Next, an AP endonuclease cuts the DNA next to the AP site, and either just that single nucleotide is removed, or a string of up to 10 nucleotides are removed. Once again, DNA polymerase fills in the gap and ligase seals it shut. The last type of single-strand repair pathway is mismatch repair, and this fixes mutations that have resulted in a DNA mismatch, which is when bases that are lined up in a complementary sequence don't match. For example, in this case, the A and the C don't match. This can happen during DNA replication, since the DNA polymerase proofreading is good but not perfect. Now, to repair this is a little more complicated than the two pathways we've talked about already, but in both of those it was easy to tell where the damage was. Thymidine dimers and covalently modified bases are obviously not normal, but in this case, it's not immediately clear which strand is normal and which is mutant. Should this A be bound to a T, or should the C be bound to a G? To handle this issue, cells can look at methylation. Some cytosines are methylated for a few reasons, including regulation of gene expression in CpG islands, and the strand that is not methylated must be the new strand, since it takes some time after DNA replicates for methylation to happen. So this is probably the one with the error, and it must be repaired. From this point, it becomes pretty similar to what we already discussed. Endonucleases will come nick the mutant strand upstream and downstream from the mutant base, then exonucleases remove that piece of DNA, and DNA polymerase comes and fills it in, and ligase seals the holes. If enzymes involved in mismatch repair are mutant or deficient, patients have a high prevalence of hereditary non-polyposis colorectal cancer, or HNPCC. This isn't nearly as bad as xeroderma pigmentosum that you get with a defect in nucleotide excision repair. Patients with defects in mismatch repair typically don't get cancer until they're in their 40s, and some will never have any serious problems. That should give you an idea of how accurate DNA synthesis usually is, since the wrong base is added so infrequently that even if you can't fix it, you might never have any problems. The last repair pathway I'll go over fixes double-strand breaks. So let's say we have this double-stranded DNA, and it gets broken apart. Well, we need to put them back together again, and this is called non-homologous end joining, because it joins together the two ends of double-stranded DNA, which are not homologous, since they are clean breaks with no overhanging end. The mechanism of this one is low yield for your exam, so I won't go through it, but the clinical consequences are important. First of all, what causes double-strand breaks? It's most commonly X-ray radiation. So patients that have a defect in one of the enzymes involved in this repair pathway would be particularly sensitive to X-rays and CT scans, and you should avoid these if possible. Also, the VDJ recombination pathway, which is used by lymphocytes to make random immunoglobulins and T-cell receptors, uses non-homologous end joining. So patients who are deficient in this pathway can have severe combined immunodeficiency. I also want to mention ataxia telangiectasia. So ataxia telangiectasia is caused by a defect in the gene ATM, which stands for ataxia telangiectasia mutated. And guess what this gene does? It's involved in non-homologous recombination. So if ATM is mutated, you get this pretty severe disease that includes ataxia, which is basically poor coordination, telangiectasia, which are dilated blood vessels near the surface of the skin, and immunodeficiency, since you can't do VDJ recombination. DNA, RNA, and protein synthesis direction is not particularly high yield, but I'll go through the important parts. You should definitely know that both DNA and RNA synthesis proceed in the 5' to 3' direction, and proteins are synthesized from the N to the C terminus. It might help you to think that 5 to 3 rhymes with N to C. So remember, 5 to 3, N to C. Now what do 5' and 3' even mean? These numbers correspond to the carbons in the sugar, with the 1 being closest to the base, going all the way around to the 5 being next to the phosphates. Aside from that, you should know that the nucleotides are added to a growing strand by having the 3' hydroxyl group in the last nucleotide in the chain bind to the first phosphorus in the nucleotide triphosphate, which is attached to the 5' carbon. This leaves you with 1 phosphate group connecting each pair of nucleotides. This is important because some drugs that block DNA synthesis are basically just nucleotides that have modifications to this 3' hydroxyl group. So they're still able to be added to the DNA strand, but don't allow it to grow further. For example, the thymidine analog zidovudine, also called AZT, is used to treat HIV because the viral reverse transcriptase will add it to the growing strand, but then can't build off it any further, since it's missing the 3' hydroxyl group. This azido group helps it cross the lipid bilayer to get into cells. Stavudine is also used to treat HIV using the same mechanism, only instead of replacing the hydroxyl with an azido group, it's just been removed. Okay, on to our next flash quiz slide. What type of DNA repair fixes damage caused by UV light? So this one would be nucleotide excision repair, and remember, someone with a deficiency in this repair pathway would have xeroderma pigmentosum, and would develop skin cancer really easily when exposed to sunlight. Okay, you're probably getting sick of DNA by now, so let's move on to RNA. mRNA is basically used to send a message from DNA to the ribosome so a protein can be made. This figure shows a typical layout of an mRNA molecule. We'll come back to this later, but for now I want to point out that the coding region, shown in orange, is where the codons are, but there's actually a lot more to an mRNA molecule than codons. There's a specific codon that designates the start of the coding region. Do you know what that is? That's AUG, which codes for methionine. In prokaryotes, this codes for formylmethionine. In very rare occasions, it can also be GUG. The stop codons, UAA, UAG, and UGA, don't code for any amino acid, but instead are recognized by proteins called release factors and result in the termination of translation in the ribosome. Alright, let's take a step back and see how this messenger RNA came from DNA. Keep in mind that the translation start site, which is the AUG codon right here, is not the same place as the transcription start site, which is the beginning of the 5' untranslated region. And the same is true at the other end. There's a lot of stuff that's translated into mRNA that will not be in the protein, which ends here. What are these untranslated regions for? They actually have a lot of important functions, like they have instructions for modifying the ends of the mRNA to add the 5' cap and the poly-A tail. And the 5' UTR has basically a landing strip that guides the ribosome to dock on the mRNA. There are also other sequences within these UTRs that are important in regulating where the mRNA ends up in the cell and how long it lasts. Okay, so that's the mRNA. Now the genes, as in DNA, have a few parts to them as well. At the 5' end, there are promoters, enhancers, and silencers. These are just sequences within the DNA, not other types of molecules. Proteins like transcription factors can recognize these sequences and bind to them to activate transcription in the case of promoters or enhancers, or repress transcription when they bind to silencers. Generally, promoters are within a few hundred base pairs of the transcription start site, whereas enhancers and silencers can be tens of thousands of base pairs away and can also be found in the gene itself, mostly in the introns. One of the most important sites is the TATA box, which is an area about 25 nucleotides upstream from the transcription start site in eukaryotes. Why is it called the TATA box? Because it's a sequence of mostly T's and A's. Now, if you remember the beginning of the lecture, is a sequence of lots of T's and A's going to melt apart easily, or are these T's and A's going to hold tightly to each other? So the answer there is they're going to melt apart easily. And this is important because the TATA box is where the proteins involved in transcription begin assembling, since they can easily separate the two strands of DNA here. In fact, the first protein to start the process of transcription is called the TATA box binding protein, or TBP. It can bind here and recruit other transcription factors. There's also a similar sequence called the CAT box, which is about 75 base pairs upstream from the transcription start site. And this also helps regulate transcription. Okay, so the transcribed region, which is this here, is made up of introns and exons. Which have the part of the mRNA that will get translated? Right, that would be the exons. The exons are the part that will actually make it into the final mRNA, and the introns will be removed by a process called splicing. Once again, the transcription start site here, which will be the beginning of the mRNA, is different from where the start codon is, since the start codon is the start of translation, and it codes for the first amino acid of the protein. If we compare this DNA image to the RNA image above, transcription begins here, and then we have our 5' untranslated region, and the start codon is further down. Another thing I want to point out about this diagram is that the 5' cap and 3' poly-A tail are added later, and were not part of the original DNA sequence. Okay, the last thing for this slide, make sure you keep in mind the difference between the template strand and the coding strand. Remember, we have our two strands of DNA that are complementary to each other. Which strand is more similar to the mRNA, the template strand or the coding strand? Well, the template strand is read by RNA polymerase to make the RNA, like as its template, so it's complementary to the mRNA. The coding strand will have almost the same sequence as the mRNA with two exceptions. Do you know what those are? The mRNA is only different in that it has uracil instead of thymine, and it has the intron spliced out. And to review from earlier, if mutations occur in promoters, it can change the amount of transcription of that gene, usually by reducing it. I already talked about enhancers, silencers, and promoters, but I want to point out one more thing about how they work. Promoters, which are the region 1 or 2,000 base pairs before the transcription start site, are relatively straightforward. Proteins like the TATA binding protein, or TBP, can recognize and bind them, and recruit RNA polymerase and other proteins to help out with transcription. But enhancers are thousands of base pairs away. So how do they work? Well, the way I've drawn this is a big clue, but they serve as docking sites for other proteins, and ultimately the whole DNA strand can bend around, so they can help regulate transcription too. There isn't a whole lot you have to know about RNA polymerases, but here are the key points. The easiest way to keep these straight is that they're numbered in the same order that they're used in protein synthesis from the perspective of the ribosome. Type 1 makes rRNA, which is the first RNA in the ribosome. Type 2 makes mRNA, which gets to the ribosome next, and Type 3 makes tRNA, which gets to the ribosome last. Type 2 is what opens DNA at the start of transcription, and none of them have any proofreading ability. Also, prokaryotes only have one type of RNA polymerase, which does everything. Alright, so speaking of prokaryotes, there's an antibiotic which targets the bacterial RNA polymerase, which is rifampin. Do you remember what rifampin's used for? It kills mycobacteria, both Mycobacterium tuberculosis and Mycobacterium leprae, which cause tuberculosis and leprosy. A second drug that targets RNA polymerase, but this time both eukaryotic and prokaryotic, is actinomycin D, or dactinomycin. What's this used for? Well, we're broadly inhibiting transcription in our own eukaryotic cells, so this is actually used as a chemotherapeutic agent. In particular, it's used to treat Ewing sarcoma, rhabdomyosarcoma, and metastatic testicular tumors. I also want to mention this toxin called alpha-amanitin, which is found in deathcap mushrooms. It inhibits RNA polymerase type 2, which probably affects most organs and systems, but clinically manifests as hepatotoxicity. Alright, for our next flash quiz slide, can you tell me what amino acid is encoded by the codon AUG? This would be methionine, which is the start codon, although this can be used as a normal codon as well, not just at the start.